And excellent. And thank you all for coming along today. I really appreciate it. I certainly know and feel it myself that Zoom fatigue is definitely kicking in at the moment. So I really do appreciate the sort of time you've taken to join us this morning to learn more about our product portfolio. Just a quick introduction to Millennium Science, who are our distributor for 10x Genomics here in Australia. And then, of course, we have myself, who is a direct employee of 10x Genomics, a science and technology advisor. Pretty new to the role, but certainly not new to the company. I've been with 10x for many years now and was until recently located in the US, where I was within the R&D program building all these cool products you're going to see today. So it's good to be back in Australia, and I'm really excited to be talking about our product portfolio. So for today, this is an overview of what I got to discuss, a quick introduction to our product portfolio, and then we'll discuss our Chromium single cell platform, our special transcriptomics platform, and then conclude with sort of our Speak with a 10 Expert initiative that we've kicked off, which gives you the ability to connect with us directly at 10x and talk about your projects or anything single cell or spatial transcriptomics related, really. So let's get started. We'll begin with our product portfolio introduction. So our assays can be broadly associated with one of three major technology platforms. We'll begin with our single cell platform. As you can see, this consists of an instrument. It's called the Chromium controller, and it's about the size of a shoebox, and it fits on your lab bench. And into this instrument, you feed these disposable microfluidic chips, and then onto those chips, you can run one of four main assay types, and I'll dive deeper into these in the next few slides. But in the meantime, I want to highlight the sample format that's required here, which is a suspension of single cells or single nuclei into this platform. These, of course, can be derived from cells that are already in suspension, say cultured cells or PBMCs. Otherwise, it requires you taking your tissue of interest and dissociating it into single cells or a single nuclei suspension. The second major technology platform that we have is our Visium spatial transcriptomics platform. And unlike our Chromium single cell platform, here there's no instrument involved. So now you're just dealing with these consumable glass slides that contain a barcoded arrays on their surface. And we'll dive more into those later on too. Also, unlike the Chromium platform, this technology now uses whole tissue slices as the input rather than a suspension of single cells. So just real briefly, here the tissue is placed on the slide and transcripts are diffused from within the tissue down to the surface of the slide, where we can barcode them on this array that's on the surface. And then following sequencing, we identify what the transcript is, and then we use this barcoded or this barcode that's appended to the transcript, place the transcript back into a 2D grid, which gives rise to the spatial context to the transcript. And we'll cover Visium in more detail in the second half of this presentation. But let's move to our third major technology platform, which is our in situ product. And this piece of technology is something that I personally find very super mind-blowing. And just like the Visium platform, here the input into this workflow will be tissue sections. However, onto this platform, you're now detecting mRNA molecules directly in the tissue itself with cellular or subcellular resolution. So rather than extracting the mRNA out of this tissue, barcoding it on a 2D array, sequencing that barcoded transcript, then use that barcode to place the transcript back into its spatial context, here you're just detecting the presence of the mRNA molecule within the tissue itself. That gives rise to data such as this. Here's an outline of a mouse brain showing the entire image tissue. The color is a powerful representation of how rich the data is, and this becomes even more apparent when zooming in and focusing on specific cellular markers. Here at high magnification, you can appreciate the single molecule resolution of the in situ array, with each symbol here representing an individual RNA molecule. However, since this is still a product that's under development, I won't be talking about it beyond this example today. But if you do kind of want to know more about this or want to keep abreast of the information as it comes to hand, please reach out. But for now, let's refocus our attention back to the Chromium single cell and Visium spatial transcriptomic platforms and briefly step through each of these assay types. And we'll begin with our Chromium single cell platform. So as I mentioned, it's about a shoebox sized instrument. It sits on your lab bench, and into that you feed these disposable microfluidic cartridges, and onto those cartridges you can run one of four main assay types. And then there's a whole bunch of bolt-on extra features that you can run within some of these assays. Because it's the same, I guess, fundamental microfluidic cartridge that is run across these different assays, each of them generally has the same high-level workflow. That's where you begin with your suspension of single cells, you load that onto the chip along with some other reagents that goes in the instrument. We'll explain more about that process on the next slide. Following that is some sort of biochemical incubation to append barcodes to your analytes of interest, and barcodes are sections of DNA. We then sequence these libraries using your Illumina short-read sequencing platforms that you've either already got in your lab or at least have access to, and then analyze the data. And we have two key pieces of software that are freely available that you can download from our website. We have Cell Ranger, which is an analysis pipeline, and that takes the raw sequencing data and converts it into a bunch of usable output files. And then we have our Loop browser software, and this is a visualization tool. So you can take one of the outputs from your Cell Ranger analysis, load it into this Loop browser, pop up the T-SNP or UMAP projection of your results, and really start to dive into your biological question of interest instantly. You start to do differential gene expression, reclustering, aggregating samples, all sorts of things. It's really quite a powerful tool. And we also completely recognize there are also a bunch of community developed third-party tools that are also equally as powerful. So the outputs of our Cell Ranger pipeline are definitely compatible with a bunch of those. But the key take home here is that we've got ready to use robust workflows, including demonstrated protocols, various sound types, and turnkey software tools for all of these solutions here. So let's take a little deeper dive into what's actually happening on this microfluidic chip. And again, this process is common across all 4 of these assay types. So you begin with your sample or suspension of single cells, and you mix that with some biochemical reagents, and that gets loaded onto the chip. You take barcoded gel beads, then that gets loaded onto the chip. And then you load some partitioning oil. The chip gets placed in this instrument, and you just push a button on the front of the instrument and walk away. And over a process of about 18 minutes, this is what's occurring so inside that instrument on that microfluidic chip. So you have your sample that flows through one channel, and you have the barcoded gel beads that flow through another channel. And then where they meet in the outlet well, a droplet is formed, or what we call a gem. Inside each gem, you'll see a single barcoded gel bead is present. Importantly, that barcoded gel bead contains billions and billions of copies of the same barcode, but then each droplet has a different barcode in it. And then if we zoom into a droplet that contains, or a gem that contains a single cell, what happens is the cell lyses and releases its nucleic acid contents, importantly, into just the confinements of that droplet. The gel bead dissolves, and the primers and the barcodes from those gel beads are released in solution. And then we append barcodes to our analytes of interest within that droplet environment. So if we're doing single cell gene expression, we're after mRNA transcripts. So we do a reverse transcription step in this, where we append, again, a barcode to our transcript of interest. And then eventually, when we sequence these molecules, of course, we use the sequence to identify what the molecule is. So if we're back to working with transcripts, we're identifying what that gene is that the transcript came from. And then we also identify what is the barcode that's appended to that transcript. And then we group all the same barcodes together and say they must have come from the same single cell or from the same droplet. Because it's the same process happening across all 4 of these assays, we have sort of high-level specifications that are applicable to each of these assays. So we have our standard throughput kits, and each of these assays here come in sort of the standard throughput format. And then we've just recently launched high throughput versions of just the single cell gene expression and single cell immune profiling kits. And this allows you to scale throughput in sort of 2 separate ways. And we'll sort of step through that now. So our standard throughput, we have 8 channels per chip, and you can run anywhere from 1 to 8 channels. And then our high throughput, we have 16 channels per chip, so double the number of samples you can run on a given chip. For our standard throughput, you can run anywhere from 500 to 10,000 cells if you per lane. And then on our high throughput, doubling that again. So you can run anywhere from 2,000 to 20,000 cells. So you can see we'll double the number of samples you can run at once, and then double the number of cells you can have on a per channel basis. So a total 4x increase in throughput for our high throughput versions of those 2 assays I just talked about before. You can also push the limits of our system beyond these cell throughputs here using what we call cell multiplexing, and I'll discuss that sort of in some later slides. But essentially, you apply an upfront oligonucleotide to your samples. That allows you to pool multiple samples together, and using that oligonucleotide sequence, you can identify those samples informatically, and it allows you to sort of load more cells in a given channel. So you can get up to, in a standard throughput, up to 30,000 cells per channel if taking this approach, and then a high throughput up to 60,000 cells per channel taking this approach. We have a multiplet rate of 0.8% and 0.4% for standard and high throughput respectively. And what that really means is that's their droplets or partitions where 1 or 2 or more cells are co-located in there. And then a cell capture rate of up to 65% for both throughput levels. a lot of people ask, well, where do the cells go? Why is it only 65%? And that's really just dead volume losses. So you begin with your suspension of single cells, you load that on the chip, so you don't load all of that onto the chip, so there's some left behind in the tube, so there's losses there. And then what we load on the chip, all of that isn't carried forward into subsequent biochemical steps. So there's a little bit left over on the chip, so that's where our dead volume losses come from, and that's where our cell capture rate comes from, up to 65%. So up to 65% of what you put into this tube at the beginning of the workflow will end up as sequenceable libraries at the end. So I just want to step through now each one of those assay types and give you a flavor for sort of how they work and the analytes you can detect with them. And we'll begin with single cell 3' gene expression. So the target analyte from the solution is mRNA, where we're capturing transcriptome-wide 3' digital gene expression from single cells. And that's definitely the core of the assay, but we can actually extend the menu of analytes being detected from a single cell using what we call our feature barcoding technology. So you can also detect cell surface proteins in addition to transcriptome-wide gene expression from the exact same single cell. This is achieved by having DNA barcoded antibodies which bind to cell surface proteins. And it's really akin to FACS, where in FACS you use a fluorophore barcoded antibody to detect cell surface proteins, but now we're using a DNA barcoded antibody. Because we're using this DNA barcode rather than a fluorophore, the number of cell surface proteins that can be multiplexed is essentially unlimited, as you're now not limited by the spectral overlap of dyes. If you're using FACS, you're only running a handful of cell surface proteins at once. However, of course, there are some, in reality, practical limitations to how many cell surface proteins you can simultaneously detect. But we have a third-party vendor and a commercial partner in the form of BioLegend who sell pre-titrated antibody panels, and you can buy antibody panels of over 100 antibodies specifically for cell surface proteins, and we routinely see this being used in the literature. So, FACS your handful of cell surface proteins, maybe 4 or 5. Here in this feature barcoding approach, 4 or 5 is easy, 30, 40, right up to hundreds of cell surface proteins at once. And again, that's simultaneous measurement with whole transcriptome gene expression. Using feature barcoding, you can also detect CRISPR perturbations alongside transcriptome-wide gene expression from the same single cells. And then, in fact, you could even layer on cell surface protein into that as well if you wanted to. And the way this CRISPR application works is you incorporate a small section of DNA into the guide RNA so that it can be captured by the assay biochemistry. So then ultimately what you get is a readout of what the guide RNA was, which tells you about the CRISPR perturbation that occurred in this single cell, but you're also getting transcriptome-wide snapshot of the functional impact of that perturbation. So you know what the guide RNA, you know what the perturbation was, and now you can look at transcriptome-wide impact of that perturbation. Was my gene knocked down, upregulated, downregulated, and then what were the networked effects of that individual CRISPR perturbation? So the single cell gene expression kit has clearly been the workhorse for most researchers and has a place in almost all areas of biology. We've seen this kit be used across a variety of different applications, really from tissue atlasing to studying the tumor, the microenvironment, right through to sort of many more applications. And here I've just taken a snapshot of some key papers where single cell gene expression has been used to make an impact within pediatric research, since I thought that would be most relevant to everyone here today. Rather than dive into each of these examples, I've just picked one to cover in a little bit more detail. And that's this paper here, where the authors used single cell gene expression kit to reveal stalled development programs are actually at the root of pediatric brain tumors. We won't dive deep into this paper, but at a high level, they generated single cell transcriptome atlas of over 65,000 cells from embryonal pons and forebrain. Then using this atlas, they derived signatures of 191 distinct cell populations and defined their regional cellular diversity and differentiation dynamics. Their findings identified impaired differentiation of specific neural progenitors as a common mechanism underlying pediatric cancers, and now provide a rational framework for future modeling and therapeutic interventions. So again, we're not going to dive deep into this biology of this paper here. We really don't have time. We could spend a whole presentation just on this. But I think it's a really great example of how single cell gene expression was used for both atlasing purposes, but then ultimately to derive deep biological insights into, honestly, what's a pretty awful disease. Moving on to our single cell immune profiling kit. Just like our previous example, the core of this assay is detection of mRNA molecules, but now we're capturing whole transcriptome 5' digital gene expression from single cells rather than 3'. So we're at the other end of the transcript now. Because we're capturing 5' ends of the transcripts rather than 3', we can expand the menu of analytes to now include full-length paired VDJ sequences from T and B cells. And the way this works is pretty simple. You perform a PCR enrichment step from whole transcriptome mRNA molecules that were captured from single cells to essentially pull out the VDJ sequences. So this means there's no computational wizardry here to do the pairing of the alpha beta chains if you're looking at T cells, or heavy and light chains if you're looking at B cells. You're detecting exactly what the cell is making, so the pairing information comes from the cell itself. So alongside whole transcriptome and full-length paired VDJ sequences from the same single cell, you can now use feature barcoding to expand the menu of analytes even more. There's, of course, cell surface protein information that can be captured in this assay too. And then whilst the oligo barcodes that are added to the antibodies for this assay are different in the structure compared to the 3' example I showed before, meaning the antibodies you would order for 5' assay aren't applicable to the 3' assay, they're not interchangeable. Basically, the barcoding premise is still the same. What is super cool now, though, is that you can, on top of all this information, layer on antigen recognition. So just like other feature barcoding assays, this works by applying an oligo barcode indirectly to your antigen of interest, so that when the antigen binds the T cell or B cell surface receptor, it remains bound, and this oligo that is present can now be picked up by the assay biochemistry and used to infer the molecular identity of the antigen that's binding your surface receptor. To me, it's honestly pretty mind-blowing. It can capture full-length paired cell surface receptor sequences and identify the antigen that it binds to. So from just one assay, you can get whole transcriptome digital gene expression, full-length paired VDJ sequences, cell surface protein, and antigen recognition. Again, that's all from the exact same single cell, but at a scale of thousands, right up to even millions of single cells at a time. To demonstrate the power of the single cell immune profiling kit and the ability to simultaneously measure so many aspects of the same single cell, here's an example of just some data that we generated in-house at Genomics. We took a pool of PBMCs and stained them with total seq-C antibodies, which come from our commercial partner BioLegend. In this panel, there are antibodies that can bind 14 different cell surface proteins, as well as some isotype controls thrown in there as well. We then ran these stained PBMCs through our immune profiling kit on the Chromium controller and obtained gene expression, full-length paired VDJ profiles, and cell surface protein information, all from the exact same cells, but of course at a scale of thousands of cells at a time. So the first piece of data I want to examine is a resulting t-SNE projection of just the gene expression information that's shown on the left. You can see we can broadly classify our major subpopulations of cell types that are present in our PBMCs. And then we move to the right-hand side now. What you're looking at here is the exact same cells, but now we've clustered them based on that cell surface protein information that we also collected. So there's 14 different cell surface proteins. And then to help us further annotate these cells, we took what we learned from the gene expression and then also layered it on here. So in doing so, we have a deeper readout and able to classify more sub-cell types from our starting PBMC population. And you can sort of see here how we're starting to tease apart now different T cells subpopulations. And it really highlights the importance of measuring two different analytes from the same single cell, both protein and RNA. The reason protein is useful, protein can be high concentration on the cell surface. It can be a longer lived species. And it can also reflect different cell states other than what the mRNA is doing. We quite often find that what we're detecting at the mRNA level can be different to what we're detecting at the cell surface protein level. And of course, always lowly expressed transcripts can be difficult to detect at RNA level, but at the protein level, we can clearly identify them. And combining both those measurements from the exact same single cell here, again, allows us to dive deeper into our population of PBMCs and tease apart the different cell types and the different biology that's happening. We're not finished there, of course, for this exact same population of single cells. And here's our gene expression t-SNE again. We can pull out full-length paired VDJ sequences for the T cells, so the paired alpha-beta chains. So the blue dots here represent single T cells where we're able to pull out a full-length paired VDJ sequence for. Over on the right, you can see our top clonotype plot. So we measure the clonotypes. And on the left, you can see just for one of those clonotypes what the alpha and beta sequence actually is. And again, we're detecting this at the mRNA level, so that's our output here, but we can easily convert that to the protein level. And then, of course, you get that same information, but for B cells as well. So again, here, our blue dots represent individual B cells where we've pulled out full-length paired VDJ sequences. The right-hand side is our top clonotypes that we've detected, and the left-hand side here is just pulling out the heavy and light chain for one of those clonotypes. Again, it's at the mRNA level, so we have an mRNA sequence here, but you can convert that to protein pretty easily. So just to really hammer the point home again here, from the exact same single cell, you can get whole transcriptome gene expression, cell surface protein, and then pull out full-length paired VDJ sequences. And then on top of that, you can also layer on antigen recognition that I didn't put here in this example, but for those T cells or B cells that you've identified the full-length VDJ pair for, you could throw antigens in there that are barcoded to also find out what their cognate pair of antigen is that they're actually binding to. So just like our single cell gene expression kit, single cell immune profiling has also been used across a variety of different applications out there in the literature. So for today, I just took a quick snapshot of a paper that I thought was really relevant again to the audience today. And in this paper, which is entitled Mucosal Profiling of Pediatric Onset Colitis and IBD Reveals Common Pathogenic and Therapeutic Pathways, the authors used the Chromium single cell immune profiling solution to understand cellular interactions that contribute to disease and identify novel therapeutic targets to answer their question of what cell types are responsible for disease, essentially. Here they profiled epithelial cells from a cohort of 17 patients using the Chromium single cell immune profiling kit to look at whole transcriptomes and full-length paired VDJ sequences from both T and B cells. The authors note in their publication that they use single cell as it was able to deliver insights from heterogeneous populations of cells that bulk RNA sequencing from the same populations of flow-sorted cells just could not deliver. Digging into this paper a little deeper, single cell provided a transcriptional landscape of colonic mucosae for children with colitis and IBD. From this, the authors then identified candidate risk genes that might promote disease development. Based on that, they identified that across the diseases, the pathogenesis is a decreased CAMP response pathway, so sort of a common pathogenesis. And then based on all this information, they then identified a therapeutic target and its associated drug in the form of dipyridamole, which is a PDE inhibitor with good safety record and wide availability. So then they showed that this drug can successfully ameliorate clinical symptoms in both mice and, importantly, in all the 9 children with colitis that were in this study. So you have a prime example of using single cell to investigate cellular interactions that contribute to disease, which lead to identification of novel therapeutic targets and ultimately a very promising drug candidate and outcomes in this example. We'll switch gears a little bit now and talk about our single cell Ataxic assay. So now instead of mRNA, we're actually detecting genomic DNA, and we're doing that in the form of open chromatin regions. For those of you familiar with the Ataxic assay, this is just the single cell version of it, where we take a transposase and apply it to nuclei, and only regions of open chromatin are accessible to this enzyme, where this enzyme then cuts that DNA or inserts itself into that DNA to make fragments of it, which we can then barcode in subsequent steps and get a single cell readout. This is our first foray into the world of epigenetics, and the open chromatin information reveals areas of active gene transcription, regulatory regions, and binding site motifs. So again, just another quick example of some data that we generated in-house at 10x Genomics. We're looking at 10,000 PBMCs here, and we can use this Ataxic signal to do clustering on our cells in the same way that we use that, or in a similar way that we use our gene expression information to cluster cells. So here, again, this is open chromatin regions that have led to a clustering of our major subcell populations from our PBMC example. But unlike gene expression, we can dive a little bit deeper into our data because gene expression, we're just detecting is a gene present or absent. But here we're looking at the regulatory environment around a gene, or the peaks that are present around a gene. So we use CD4 as an example here. So again, if we're doing just at the mRNA level, we would count, is CD4 present or is it absent from this cell? Is it a 1 or a 0? But from the Ataxic, we can look at the peaks centered around this gene. So we find a promoter accessibility peak in these cell types for this gene. We also find a repressor for the monocytes and DC cells. And then we actually find an enhancer for the CD4 cells only. So again, it allows you to take a deep look into the regulatory environment around a gene, not just count is the gene present or absent, and can really help you sort of classify your cell types and really understand what they're doing at the epigenetic level. So we'll finish this with our Chromium single cell multiome. Really, the moment we launched our Ataxic assay, everyone understood the power of that Ataxic measurement that I just went through, but also immediately jumped on, well, what if I could get both epigenetic and whole transcriptome information from the exact same single cell? I want the open chromatin, and then I want the whole transcriptome mRNA I want to know what both those things are doing, but I want it from the exact same cell. I don't want to run both kits in parallel and then infer. And that's where this product is born from, our Single Cell Multiome Kit, which is really an amalgamation of our Ataxic and 3' gene expression assays. It allows identification of putative regulatory elements, and you can then directly link them to a gene of interest from the exact same single cell. So open chromatin and mRNA. If we look at another example of data that we generated in-house at 10x using PBMCs again, and you can see PBMCs as one of our favorite sample types within R&D at 10x. We're looking at the gene expression element of our data set now, so whole transcriptome-wide 3' gene expression, and again, we do our clustering here to broadly identify our major subcell populations that are present in PBMC. Here's our Ataxic signal from the exact same cells. Here's our open chromatin profiles that again allow us to broadly classify these major subcell types. But what we can do now is using the barcoding information that's appended both the transcript and the genomic DNA molecule, we can use that barcode information to link these two signals together. So we call this inference-free linkage, but what we're measuring again is transcriptome-wide gene expression and genome-wide chromatin profiles from the exact same single cell. So we know they're coming from the same single cell based on that barcoding, them containing the same barcode that's from that barcoding step that I sort of talked about earlier. And as a quick example that I think nicely highlights the power of simultaneous attack and mRNA measurements from the same cell, I've selected this work here from the researchers at Mount Sinai. Here they use the Multiome Kit to deeply study a population of human pituitary stem cells, which are important in tissue homeostasis and renewal, but have been incompletely characterized. What you can see in that UMAP plot here is that the clustering of different cell types identified in the human pituitary gland based on their combined gene expression and attack signals. So this is the multiome clustering over here on the UMAP plot. And then since these researchers were primarily interested in the stem cell population, they then reclustered just these cells. And that led to the UMAP plots along the bottom. Here, where at the mRNA level, they observed differential gene expression patterns in the apparently heterogeneous stem cells. So just because cells are the same type doesn't mean that they express exactly the same set of genes. But the key question is, what are the mechanisms that are leading to differential expression within this stem cell population? So we'll quickly examine how they use the multiome data to provide insights into transcription factors and epigenetic mechanisms that shape gene expression within the same pituitary stem cell type. Firstly, up the top here, we can examine the GATA1 gene, where authors actually found no change in transcription factor expression between stem cells. So there was nothing at the mRNA level that was telling here. However, when they did observe significant changes in chromatin accessibility across stem cells as highlighted by the red bar. So at the mRNA level, they weren't detecting anything, but at the chromatin level, they could clearly see a difference. And this suggests that the chromatin accessibility is a predominant driver of the GATA3 differential expression within stem cells. The next example is the POMC gene. And unlike the previous example, here, there was actually no change in chromatin accessibility. So you're not finding anything at the chromatin level. However, at the mRNA level, there were significant differences in the expression of two key transcription factors that are shown in that blue box there. So this suggests that it is differences in the transcription factor expression that's a predominant mechanism of this gene within the stem cells. So the acquisition of both the mRNA and open chromatin data from the same sample provided high-resolution analysis of the chromatin accessibility pattern of key genes within the pituitary system cells and revealed potential novel regulatory domains. And again, really nicely highlights why you want to measure both of those modalities in parallel from the exact same single cell. So I'll round out this section with new products that we have coming for our Chromium single cell platform. So available now, launched this year, we have targeting panels, Cellplex, and our low throughput kit. So targeting panels are exactly what they sound like. You still generate your whole transcriptome gene expression information from our 3' and 5' kits, but now rather than sequencing that whole transcriptome, you might only be interested in a subset of genes. So rather than wasting sequencing money, we then sell targeting panels in the form of pre-designed panels, or you can design your own. We just pull down the genes you're interested in and only sequence those. So it saves some money on the sequencer. We have Cellplex Reagents, and this is our sample multiplexing that I touched on earlier. Here, you're not saving money with the sequencing, but you're definitely saving money with upfront 10x costs. So what this allows you to do is take multiple samples, apply this reagent, which is a lipid molecule with a DNA barcode on it. That lipid molecule incorporates into the membrane of cells. So you can apply different tags to each sample. You can pull all the samples together, run them as a meta sample, if you will, through one of the lanes of a 10x chip, rather than running across multiple lanes. And then you can use your informatics to computationally demultiplex the different samples based on the different tags. And again, it saves you money if we're having to run multiple 10x lanes. You can kind of put everything into one. We have a low throughput kit, which, as it sounds, limits the number of cells you can get out of the kit only down to 1,000. And the intention here is if you've got some upfront sample prep optimization you want to do and you don't want to commit to using sort of the larger scale kits, you can buy a cheaper low throughput kit. You do some whatever you need to do to be confident that you can scale up your experiment to the standard and higher throughputs. We just launched our Chromium X instrument. So this is actually a replacement of that Chromium controller that I showed you earlier. It's not very big again, it still sits on your lab bench. But what this allows you to do is run all the assays I just described, but now you can also run the high throughput versions of the 3' and 5' assays. And then coming sort of into early next year, we have fixed RNA profiling kit, 5' CRISPR, and now barcode enabled antigen mapping. So fixed RNA profiling kit, this is very exciting. What it allows you to do is generate a suspension of single cells and then fix it so that you can store those cells. So it's a proper fixation. And then you can store those cells for a period of time. And when you've got enough cells or enough samples, you can batch process them together on a chip. So it's really important if you're doing time course studies, for instance, or you're somewhere that you can't instantly get your cells onto an instrument, you can just fix it and run it later. We have 5' CRISPR, as the name suggests, we're just within that context of the immune profiling assay, also going to enable the detection of CRISPR perturbations, or more importantly, the guide RNA. So you're going to be getting whole transcriptome wide gene expression, full-length paired VDJ sequences, cell surface protein, antigen mapping, and you can also now do CRISPR detection within that assay. So again, just opening more possibilities there of what you can do with that tool. And then barcoding enabled antigen mapping, or beaming is what we call it. And this is really our commercial solution to that antigen mapping that I talked about before, where we have protocols, reagents, and software to enable you to associate antigens with their T cell or B cell surface receptors. So you bring the antigen to this product, and we supply everything else within that workflow to get you up and running. So with the final part of the talk, I want to switch gears now to our second product platform, which is Visium Spatial Gene Expression. So a lot of people ask, well, why spatial analysis? And really, location matters. So I think this is a good example to explain that. So we're looking at here a hot tumor that has good prognosis versus a cold tumor that has a poor prognosis. And we're seeing on the left the H&E staining of this tumor, on the right some immunohistochemistry of the lymphocytes. So at the top, you can see for our hot tumor, which is good prognosis, the lymphocytes are able to penetrate into that tumor and do their job. But on the bottom, a cold tumor, poor prognosis, you can see the lymphocytes are located in a ring around that tumor and unable to penetrate, and that's what leads to that poor prognosis. So of course you could run single cell gene expression here, or one of those single cell products I just talked about, but that involves dissociating this tissue into a suspension of single cells. So you lose all this spatial context. So you might be able to infer some differences between the lymphocytes on the top versus the lymphocytes on the bottom, but you just, at the end of the day, can't piece back spatially where they came from and get a picture like this. So that's where our Visium product is really born from. So this is the data types you get out of the Visium product. So of course you get the histology image. You can still do H&E staining. So you can still do the annotation of your tissue, whatever classical approach you're doing now, based on microscopy, basically. Typically that's where your analysis of this tissue would end. But what we are able to do now with Visium is capture underlying molecular information that's within this tissue, so in the form of our whole transcriptome from these cells. And then, of course, what we can do very powerfully now is combine those two data types together. We can overlay our molecular information onto our visual information and really start to develop a deep understanding of our tissue types and the gene expression patterns that are driving differences spatially. So we have two products for Visium Spatial Transcriptomics. We have our fresh frozen or FFP product. At a high level, they're basically the same output from these products, but really, as their name suggests, they're designed for the different samples that you may have in your lab. So if you've got fresh frozen tissue, you can use the fresh frozen product. If you've only got FFPE tissue, we've got a solution for that as well. Both these products use the same general high-level workflow, which is you begin with sectioning your tissue onto a slide, or the Visium slide more specifically here. Use some imaging, so whether it's H&E or immunohistochemistry. So at this point, it's pretty standard histologies, things that you're already used to. Once that imaging is complete, however, then we do some molecular biology actually on the slide to barcode our transcripts of interest. Once that barcoding process is complete, we can work back in tube format, complete our next-gen sequencing library, sequence that on our Illumina sequencing platforms. And then again, we have two key pieces of software in the form of our Space Ranger analysis pipeline and our Loop Browser visualization tool. So Space Ranger takes our raw data off the sequencer and converts it into a bunch of usable output files. And then Loop Browser, we can take one of those files from the Space Ranger output, load it into Loop Browser, and essentially get that image straight away of what I just showed you of H&E or immunohistochemistry image overlaid with our gene expression profiles. And you can do a whole bunch of powerful analyses in there. And then likewise, from our previous example, also we completely understand the community is also building some pretty cool tools out there to analyze this data. So the outputs of our Space Ranger pipeline are certainly compatible with a lot of those tools. So the key take home here, just like before, is we have ready to use robust workflows, including demonstrated protocols for various sample types and turnkey software tools to get you up and running with Visium. If we take a look at this Visium slide a little closer, just in some technical detail of what's actually occurring on this slide. So we can see the slide has 4 capture areas. So you can put 4 slices of tissue on this slide. Each capture area is 6.5 by 6.5 millimeters in size. So your tissue has to fit within there. If we zoom into the capture area, you can see each is made up of about 5,000 barcoded spots, where importantly, each barcoded spot contains a different barcode. And that's identifies it spatially within this 2D grid. The spot size here is 55 microns. So of course, those of you with a keen eye already know that's not single cell resolution, that's a handful of cells, but I've got a note on resolution on the next slide. It's something we're definitely working towards. And then if we zoom into each of those spots, again, there's a lawn of oligo. I'm not going to go through the oligo structure other than to highlight the important bit, which is the spatial barcode, where each one of these spots has its own unique barcode, and we know where that barcode is laid out in the 2D grid. So that way, when we sequence a molecule and we sequence the barcode that's appended to that molecule, we can place that molecule back onto this 2D grid. So, like I said, a quick diversion or a side note about resolution. So our product in the middle here is our current Visium arrays. We have 55 micron spot size. But what we have coming next year is what we call Visium HD, and this is super exciting. It says single-digit resolution here, but we've pushed the resolution right down to just 5 micron spot sizes, so true single-cell resolution here. So that product's coming next year. I will have a quick sort of side note that if you're really wanting single-cell resolution, or you're thinking, oh, I'll wait for that to come out, kind of think about Visium as it is today, and if you would be willing to sort of dip your toe in with it with the current product. The reason I say that is because you can really have your adoption curve bring up with Visium. You still get very powerful data out of Visium, as I'll show you in a minute. And it allows you to sort of bring that technology into your lab so that when Visium HD does launch, you can kind of drop it into your lab arena. There's no, again, adoption curve here for you to learn more about the technology and sort of the workflows and the sequencing data. You can do that today with the current Visium product. And when Visium HD comes out, boom, you can get up and running straight away. So like I said, there's 2 versions of the Visium kit, fresh frozen or FFPE, and this is sort of the main difference between these 2 kits as explained on this slide. So we'll begin with our fresh frozen tissue, or the fresh frozen kits. The way this works is we place the tissue on the slide, and then after imaging, we permeabilize that tissue to make the mRNA molecules diffuse from the tissue down to the surface of the slide, where we barcode those mRNA molecules directly. So basically anything that's got a poly A tail is what we capture in this assay. Because of that, it's applicable to a whole bunch of different species. We see it used for lots of different things, including plants. People also sort of use it for that too. FFPE is a little different though. FFPE fixed tissue, of course, has the fixing process itself. It can damage the RNA molecules, cross-link them together and make them inaccessible. So rather than capturing the mRNA molecules here, what we're actually detecting are probes that are binding to the mRNA molecules. The way this works is we take our tissue slice, we apply it directly again to the surface of our slide, we do our imaging, and then after our imaging now, rather than diffusing the mRNA molecules in this tissue directly down to the surface of the slide, we actually flow probes onto the tissue. So the probes are for different protein coding transcripts. If the transcript is present, the probe binds. If the transcript is not present, the probe is washed away. We perform a biochemistry step to lock in the specificity of those probes. And then what we do then at that point is permeabilize the tissue and diffuse the probes rather than the mRNA molecules down to the surface of the slide, where now we're capturing the probes and we sequence the probes, and of course the probes represent the genes that were present. Because this is a probe-based solution, we've only got probes at the moment for human and mouse. So if you're looking to do this on sort of tissue types outside of these species, then we'll just sort of have to wait and see what's going to come out of R&D in terms of our development, what we're going to support next. But at the moment, it's applicable for human and mouse. And to finish on for today, I just want to take you through a quick story or a data vignette that we generated internally at 10x using our Visium products, both our FFPE and our fresh frozen version of Visium. So triple positive ductal carcinoma in situ is basically a precursor lesion that can give rise to invasive breast carcinoma. And the major expression signature or the hallmark of this is 3 cell surface receptors, HER2 protein, the ER protein, and the PR protein. So to better understand the tumorigenesis, we spatially profiled the transcriptome of the microenvironment using both Visium FFPE and Visium fresh frozen on the exact same tissue slice here. So we took some of the tissue, we fixed it, we applied it to Visium FFPE, and then we had the fresh tissue and we applied it to the fresh frozen Visium. So here's our H&E image, and this is actually from the fixed tissue. And then here's a comparison between the results we got out of fresh frozen versus the results we got out of fixed tissue. And again, we're looking at here, I guess the most important plot is actually on the right. Middle plot is, sorry, genes detected in one versus genes detected in another. So we're detecting the same genes, which is great. And then on the right is the sensitivity for both the assay types. So you can see that no matter whether we're using fresh frozen or we're using that fixed tissue, the sensitivity is comparable. We're really not degrading the performance of the product by using FFPE tissue, which is really important. FFPE here is all the fixed tissue is a little lower in sensitivity than fresh frozen, but I've definitely seen the other way around in different examples where the fixed actually gives you a little bit more sensitivity than the fresh frozen. But again, the key take home here is that we can use these different sample types into our Visium workflows, although there are some differences between the 2 different kits. At the end of the day, what you're detecting is going to be the same, which is really, really important. So we also took our H&E image and we had a pathologist annotate that image. And you can see they identified 4 main areas here. And then now what we're looking at is our gene expression profile or the underlying molecular profile from this tissue. So I'll begin with saying the pathologist annotations are not necessary to do the clustering here. We just force the clustering because the pathologist identified 4 main cell types. So we tell the clustering to go and identify or constrain it to 4 cell types. And that's what you see here. You can see here quite nicely that cluster number 2, the red, lines up very nicely with the cancerous regions that the pathologist has identified. So typically in that middle image, that's where your journey ends. The pathologist has identified cancerous regions. You can see them, but you don't know what's happening at a molecular level. But now, of course, we can layer on our gene expression information and capture that. We could do immunohistochemistry. Again, I said the hallmark signature of this type of breast cancer is these 3 protein expressions here. So that's what's shown down the bottom, immunohistochemistry. So we're now detecting at the protein level. And up the top is our gene expression. And we're detecting at the transcript level now those same proteins. You can see how closely the protein and transcript match up spatially. And then on the right-hand side, you can see we're no longer constraining the analysis to only identify 4 cell types, based on what the pathologist was able to identify. Here, we've done some unbiased clustering. And you can see that we can detect up to 9 different clusters now. And you can see even within those cancerous regions, there are differences at a molecular level as to what's happening. So it's really exciting that you can dive into that again at the just at the imaging level, you can tell where the cancer is. Here, you can dive into the molecular profile of the cancer and how it's changing spatially across the tissue. All around this talk out with some new products we've got coming in the realm of our spatial transcriptomics or a Visium product portfolio. We've got larger capture area slides coming, which, as the name suggests, with bigger capture area to put larger sections of tissues on. So rather than 6.5 by 6.5 millimeters, these will be 10 by 10 millimeters. The resolution is still the same, so 55 micron spot size, you just get more spots. Highly multiplex proteins coming next year, and that's really, really exciting. This, akin to sort of that feature barcoding that I explained about on our Chromium platform, we're going to enable that now in our Visium platform. So what you'll be able to do here is detect a large number of proteins simultaneously with genome-wide transcriptome information. So you get mRNA and protein from the exact same section of tissue. You can overlay it on each other, and of course, because we're in the DNA barcoding space now, rather than using fluorophores to detect proteins, we essentially have an unlimited plexi there, so you can look at tens to hundreds of proteins at once. CytAssist instrument, this is certainly not required, but definitely helps with the workflow. So what this instrument does is it's pretty neat. in that you input a Visium slide into this, but you also input a plain glass slide that has your tissue. So you no longer need to section your tissue onto a Visium slide. This instrument will pair your tissue with a Visium slide. So this is important for two reasons. One, it gives you access to perhaps a whole bunch of tissue you've already got pre-cut that's stored that you would like to analyze, but you can't resection that. Or it allows you to sort of take your tissue, do a bunch of cuts, put them on a plain glass slide, decide which one is important to you, or none might be, store it, and sort of repeat that process and really get to the point where you find a section of tissue that's you want to profile, and then you can move forward with that. And like I said, this instrument essentially pairs a Visium slide with tissue that's already on a cheap slide, puts them together, does that transfer of molecules from one slide to another, and then standard Visium workflow after that. And then finally, Visium HD, which of course we talked about earlier, but there's going to be a single cell resolution version of the product where the capture area will stay the same size, 6.5 by 6.5 millimeters. The workflow will stay the same. This will be compatible with fresh frozen as well as FFPE. It'll be compatible with the CytAssist instrument here, but it's all now single cell resolution, which is really exciting. So thank you so much for coming along today. Like I said, I really appreciate it. I completely get it that Zoom fatigue is really kicking in. So I'm glad you're able to stick us out here and really learn about our products and hopefully take home some cool information and think about how it can be applicable to your research. Just finally, we have this initiative that we call Meet with the 10 Experts. So we have Yvonne Peterman, who's a science and technology advisor directly employed for 10x Genomics. We have Catherine King, who's a field application scientist directly employed for 10x Genomics, and then myself, also directly employed by 10x Genomics. And I left my email address off here annoyingly, but through the Millennium website, you can find a link here of how to meet with us, and we're happy to set aside time to design anything, to talk about anything from experimental design to budgeting, workflow, grant writing, anything that you would like to know or need to know from 10x Genomics in order to move your experiments forward, data analysis. Feel free to reach out to us, and we're definitely here to help. So with that, I'll throw it open to see if there's any questions.
There was a question in the Q&A box, Jeff, from Joseph Wagner. RNA staining is amazing. Is it possible to stain for specific RNA amino acid mutation? RNA AA mutation. That was from just after you started, so I'm not sure which. I'm
not sure quite of the context of that question. So I think it probably would have been in the single cell, Chromium single cell realm. You can do upfront staining of cells and FACS sorting, those kind of things. You can do, as long as your cells are already in single cell suspension, you could do some sort of microscopy. But out of our platform, you're basically just detecting any polyadenylated species. Because we do actually sequence we sequence the transcript. We do at a high level gene counting, is the transcript present or absent? But you do have the sequence of transcript there. So if that is of interest, you need to pull out something specific that's in the sequence of the mRNA molecules detecting, say there's SNPs or something like that. That's possible, and there's certainly a whole body of literature and algorithms written to do that.
Okay, I'm not sure if we answered the question there.
Yeah, hopefully I did. I'm not sure. I guess another point that I sort of tiptoeing around the question as well, because I'm not sure I could fully understand it, but out of the assay, so we actually capture full-length mRNA transcripts from our single cells. So at midpoint through the assay, when you've generated all the cDNA, you've actually got full-length mRNA transcripts. And then to get them on the Illumina sequencing platform, we have to shear them down, and we lose a whole bunch of information from the transcript. And then you put it on the Illumina platform. There are a number of very clever customers who have realized, well, I'd like to just sequence the full length of that transcript. And have applied protocols or hacks, if you will, to then put it on either the PacBio or Oxford Nanopore. Interesting paper just came actually where they concatenated a whole bunch of mRNA molecules together to put onto the PacBio, so you could sort of reduce the cost of that platform there. But either way, you can sequence with base pair resolution the full length of the transcript as well. So you can also, using the sort of a hack of our protocol, so you can also sort of dive into at a base pair level your transcript as well, if that's important to you.
Okay. Yep, Joseph has come back and said very clear, so that's good.
Good. All right, there's no other questions. I'm happy to give people some time back for sure, and go and enjoy their morning.
Yeah. Okay, thanks for that, Jeff. I really appreciate it. Great presentation as always.
No, thank you all for coming along again. I really
Okay.
appreciate it.